Hello, everyone. Long time no see. Actually, I never seen you in this life. Somebody maybe is never seen me before. Welcome to the course of the cell biology. So, hope you are still good in your health and spirit in the pandemic of the COVID-19. So, in the first lecture, I just simply explain the term and also introduce to you some basic and some guideline and also some organized the class and also some chapter, some book we need to pass through the course. And the first and also introduction about a little bit about the first chapter in the book about the cell, the fundamental units of the life. So my name is Lemon Tong. So I'm from the Department of Biotechnology. So a little bit advertisement about my CV. You can access this website address to check about my research and also my information. Any kind of scientist, I also have this one. So I got a PhD in 2015 after and go for postdoc and also assistant professor in Korea. I come back in the last year. It's almost 1 year I'm working in IU. So my major is about immunogenetics. It's like applied genetics in the immunology to improve the disease resistance in animal. And I also have more than 10 years. The time I study and work abroad is about 11 years before I come back. And I also have experience with about more than 10 years in the animal cell culture, and also I worked when I was undergrad time in the plant cells culture. So if you have any question about the cells, and you can contact me. And I think I have a lot of things to discuss with you in the course too. This is some discipline to me, like introductory, like immunology, so evolutionary biology and genetics. So some model I use like cells, DNA, I'm working on cloning, gene expression, and also my focus on a new technology with DNA sequencing. When I come back here, I build a lab working using some molecular technology for using diagnostic and also for plant and animal breeding, and also working a little bit in human. I have some students, about more than 19 students in my lab right now, and anybody have work kind of good, I think. Okay. So I will outline a little bit about our syllabus. So the course, let's see, you say cell biology, but actually cell biology will focus more in the course about actually it should be molecular cell biology, because of the cell is a contract, cell is a basic unit of living form, but the structure of cells is from macromolecules. So you still remember 4 macromolecules in a cell, right? We have a DNA. So we have the nucleic acids like RNA, DNA, some stuff. And we had a protein, like making the enzymes, some kind of the protein is the molecule doing the work in the cells. And we have the lipid for the structure, and we have the polysaccharide also showing the structure and also cell signaling. And this is full course beta 009 IU. And this is a Arctic course, like requirement for another one. So I think this is very basic course. You can use this one to understand another course later, like genetics, biochemistry, or something. And using this one, you can understand more better, higher course. So we have a 3 credit. We don't have lab work. This course. And some requirement before enter the course, you have to know about biology, why basic, and we have the organic chemistry. Actually, biochemistry is you should know a lot, but don't worry, I will cover some of the just we need some basic about biochemistry in the course to understand better about molecular stuff in the course. Okay, about the description. So this course will be the foundation for the life science. So because we work in the cells, the fundamental units of the living form. So we will know everything about the cells, almost everything about the cell. So using the technology in this course, you can apply it to understand better for another course, like DNA, protein, later, I think. And we will have the full definition of the living cells. Living cells. What's a living cell? What is the characteristics of the cells? Living cells. So living cell is one. They are living, but living is one. And the cell classification, like bacteria, archaea, plant, animal, and another thing. Maybe somebody will ask about the viruses too. And but we focus on the eukaryote class, the eukaryote like plant and animal, because of the eukaryote complex, and it's also like animals is complex and very near to the human. Actually, we focus on the human cells a little bit. So based on the understanding about the eukaryote, especially in the animal or human cells, we will know better, use that kind of thing for relate with understand ourselves better, right? Ourselves better, like disease or a treatment of kind of or diagnostic or treatment of the disease, for example. And we will this group is like I said that we focus a lot in a molecular thing. So we will learn about the structure of the cell and the function of the cell, how cell work, and we'll shortly discuss about organelles of the cells, the organelles of the cell. So but I believe all of you is remember the organelles of the cell, right? Mitochondria, cell membrane, and lysosome or something. And macromolecules. So macromolecules is in biochemistry. You learn already. Some of you, I think. And you say uh, the how is a macromolecule in detail look like, and how they work together to make the cell become a living form. And we talk about the activity of the cell, like grow, divide, transport, the transport of the protein in the cells, and also the cell signaling. So how this cell can affect activity of another cell, by what? And also all of this activity in the same system in the cells. So we will learn some kind of tissue and how different cell type we will synchronize working in the organism to make the organism work. And we also talk about the genetic mutation, and I will briefly explain that's mutation in fact to the genetic disease. And sometimes we will talk about the story of cancer, very hot topic. So after the course, because is the information is quite very big, quite very big. So I cannot quite remember all the stuff in the cells in the course yet. But after on lecture, so you may have some big picture, big figure about how the cells, about the cell, and you have I think more deeper to understanding of the cell, like the foundation of the life science, classification, and molecular activity. And from that, you can understand better about the specific subject related with the living stuff. About textbook and grading, so I use this one, Essential Cell Biology 4 editor from 2014. Is the knowledge is updated, new and very new, but basic is quite not changed so much these days. So this book is writing is very good, I think. Simple to understand, and if any kind of statement of the book is kind of they have the figure and the picture very easy to understand. And my advice, my command to read the book is you need to read the title of each section in the chapter, and then looking further and look into the picture and try to explain the picture. And if you cannot explain, read the book, read the text. That is the fastest way to digest this book. So I very like this one. So actually, about the cell biology, we have a lot of books, really, more than 30 or 40 books. But I choose this one because it's simple and easy to understand. And the format of any book is same, but another book is kind of complex. And the grading, we have the midterm, we have about 30 or 35%. The final, we have 40% fixed. And we have the attendance and also assignment and presentation about 25 or 30%. But I don't know, because we lost some week. I just sit in here and talk myself with a computer like this, and we cannot contact each other. So I don't know how about the organization of the presentation. I'm thinking about that later. Okay, I move to the topic. For every week we will spend on. Actually, this kind of thing is completely fixed with the content of the book. So we have the chapter 1, Introduction of Cell Biology. Today we talk about that. And second is briefly some technology we're using for studying the cell and the future of the cells. So I almost relate with if you want to talking about the structure, we need to have some tool to look into the cells and take the picture of the cells. So the microscope is the most important thing. And the cell classification and cell RNA. So I also fast this one is quickly fast this one. And we have the cell energy, catalysis, and biosynthesis of the cell. Is a very similar with the biochemical stuff like somebody learned already. Because the cell is an isolated system, right? Cell is an isolated system, and they need to follow some rule of the universe. So the universe and any system, they need to follow the rule of energy. So any molecules, any organs need to follow that one to work. So I will remind this one in one chapter. And next is more complex. This one is the most complex in the book. It's a protein structure and function. We'll deeper try to understand about the protein in the cell. The type of protein and the locali of the protein in the any part of the cells. And you will surprise the protein in the cell is They contribute more than 50% of the cell membrane weight, and a lot of things. I mean, maybe uh, almost of the activity in the cell made by the protein. You will see. And this chapter will open for you very much how the cell works. So, for example, from the biology course before, you will see the cell will grow. The cell will be transmitting signal to another cell. The cell will be divide. The cell will be developed to another cell type. But how can they happen? All the activity will depend on the protein. And the structure and the function of each protein will be discussed in this chapter. And number 6, we will talk about the chromosome, DNA, and DNA reparation, and recombination. So this one is maybe somebody will be see very familiar because of in the course of genetic. In the second year, you already learn about this one a lot. So I just cover something you don't know. Like how about chromosome organization, even they are uncoiled. They are not decondensed. How chromosome in the cell, in the nucleus, they organize. They different with the cell. And explain. We have some interesting question about the chromosome or DNA. And from DNA to protein, how the cell read the genome. Yeah, like the transcription, and also the chapter 7, chapter 8 is quite close together, and the control gene expression. We have different cell type. And each cell type is do the specific work in the body. So different cell type have a different set of gene expressed. So in a human, we have a 3 billion base pair of our genome. This is quite big. Actually, 6 billion base pair, because we have the 1 copy from mom and 1 copy from dad. So we have 6 billion base pair around. So how many genes in our genome turn on and turn off is also the matter. And how the structure of gene. I mean, how the structure of genome, like repeat sequence. So, and chapter 9 is how gene and genome evolve. Also interesting. Genome is before DNA becomes the macromolecule storing genetic information and can transmit to the next generation. Talking about genetic. So think about some simple molecules. When the first cell comes to first molecule comes to this planet, and then that is a small molecules, and small molecules is favored by nature, and then they evolve. But from the small part to become the very big, like 3 billion base pair right now, very big. From the small to become very big, so they have a big event in the evolution road. So we will study and discuss that kind of event. What kind of event is most important for the genome? So we had a small genome and become a big genome, and we have the speciation, like some kind of gene we don't need it, we remove it, and yeah. And okay, from chapter 8 or chapter 9, before we had a midterm cover. That's why. So somebody will be feel familiar because of this resemble with what you learn already about genetic or biology. It's actually same. But from chapter 10 to chapter 15 is quite a very deeper in the cell molecular cell biology, because you see some structure you never know before, like membrane structure. You deeper learning about the membrane structure. Very useful in this one. So learning, discuss more deeper about the membrane structure. You will understand better for some biotechnology work, like For example, I want to transfer, to put some DNA into the cells, right? And I need to open the gate in the membrane, cell membrane, for that my DNA can enter. Okay. And another one, somebody interested in the drug delivery, right? Like you want to deliver this drug to that specific cells, and I need to learn about how the membrane structure, how the protein organized, and how that specific protein in the cell, specific cell type, and you can put that drug in that cell, not another cell. So understand more deeper about the cell membrane. You help you in that kind of work. Okay. Uh, Next, we go to intracellular compartment and protein transport. It's protein is made from where? Made from the ribosome, like you know it. But how is it from ribosome? They need to go to the Golgi. From Golgi, some protein will be taken out, or come back to the come to the come to another organelles like mitochondria, because mitochondria is not enough DNA for do the job in the mitochondria. They need some protein from the nucleus. Some protein encoding into from the nucleus to enter the mitochondria, or to do the work, or some kind of protein, and some kind of lipid we need to make the bigger cell membrane when the cell grow, right? Because when after division the cell small, they need to growing. So the growing, we need the cell membrane bigger for bigger cells. Lipid and a protein of the membrane is from where? Where it come from? And how from the ribosome in the ER can transfer to that place is what we need to discuss. And the cell signaling. So cell signaling is like the key and the lock. So this cell, to direct another cell do some work, they need to send a signal at the key and go to another cell. Another cell want to adapt or read the signal. They need something fixed with that key, the lock. So if they fix, they open the gate, and the cell will obey the signal. And very complex thing about this chapter. So learning this chapter, you understand how when you drink the drug, you will kind of overcome a disease or something, or you feel some change in your body if you drink the drug. Yes, by cell signaling. Understand about that. And cell cytoskeleton also, and a cell division cycle. Like animal cells, you see, is in the plant cell we have the cell wall, like cellular. So it's very hard, very rigid, and the stable structure of the plant cell. But how about animal cells? So animal cell is just a cell membrane, like a bubble, and easy to break, but it's not break yet. So because we have a very complex cytoskeleton, the bone of the cells, will have the cell become hard to break. And also the roles of that kind of the skeleton, the bone of the cell, to the cell division, divide. And chapter 14 will start through maybe short discussion about this one, because this one is you learn in genetic already. So I believe all of you is learn genetic already. And chapter 15 is quite important, and you see this on the type of the cell. We will contribute for tissue, like they living in the same environment or microenvironment, and communicate each other by signal, by some molecular connect them together, or by the organize of the cell type, different cell type. When you cut the one tissue in your body and look at it, you will see a lot of cell type, a lot of cell type in there. You have the blood cell, the white blood cell for immune cells, or you have the skin cells, you have the but a lot of cell type. Okay, that's what we finished. So after from the chapter 9 or chapter 10 to the 15, we hover in the final test. So the final is completely, as I said, completely different with the midterm. So the score may be dramatically drop this one. Okay, now we just briefly talk about the term. So what is biology? Like biology is about like life in the Greek, and logy and study of this is biology is science study life and the living organism. So the study is we need this we have the left side and living thing. So we have to looking for the structure. So we the living that thing is made by what? The function is how it works. The origin is where it comes from. The evolution from the simple form to the complex form, how it going. And distribution. So this kind of creature is from where localized somewhere in our planet. And classification. Okay, that is many stuff more. And we had a principle like cell theory here. So remember from the long time ago, the scientists don't have the tool like DNA sequencer. They don't have the very big and complex and modern microscope. They just see something and they make the hypothesis, right? And they make the hypothesis, and then through the time, so another scientist will focus on that hypothesis and they prove it, they correct or not. That is how the science goes. And then and then we talk about evolution and we talk about the gene. That is very simple and we have the homeostasis. Homeostasis. This is the very basic of the system. Because the cell is a living system, they kind of independent system. So homeostasis is a term set up the stable ability of the system they can work. And all the process in that system can work to maintain the system is become stable. And we like this one mentioned already, so the energy. And we have a classification like botany, plant, zoology like the animal, and microbiology is the microbe, and we also have a virus part of life here. So we dig something, we dissect our target like biochemistry, molecular biology, some micromolecules there, and cellular biology, how the cell look like, what they react with the signals, for example, and a cell physiology, and ecology also, the term we need to, actually we don't need to focus on ecology discourse. Oh, how about cell biology? Cell biology is we can also call it cytology. So remember cyto is the cells. So later on, you maybe touch this cyto a lot. So cyto is kind of cells. We talk about the structure, for example, the chemical and molecular component of the cells. The organelles, from the small scales to larger scale in the structure of the cell. And we have the process of the cell, how that thing works, like division, metabolism, signaling, gene expression, and cell death. So cell is a structure and a functional unit of all living organism. Because of any kind of living system have this kind of characteristic. So we go to the next thing, talk a little bit about history, the life on Earth, the highlight. I quite very like this figure. So see in here, uh, we have our planet as it was about 4.6 billion years ago, but this number is not very correct. So some talk about 4 billion, some talk about um, 5 billion, and some recently some think is that is wrong. It's maybe longer than that. So just from the rock, and then after that, some molecules appear and become the simple molecules, the simple molecule from the atom in our planet, and we had a molecules, and then the first living thing about 3 billion years ago. They need 1 billion, 1 or 2 billion years to make the first cells. First cell. We have a picture of this one, and then we have the nucleus. I mean um, we have the kind of DNA or some genetic transfer about after 2 billion years, and you see that from that, like that the nature mother, she have the she have something to favor. So that kind of living thing is dominant in the world, in the planet. Like you see the dinosaur in here, some simple thing, and we have some animal, and before that we and right now we have the human, like the king of the our planet. So if you compare the age of our planet in one day, so the time of the human in the world just 2 seconds. You see it, evolution is really really nice, right? A lot of stuff only happen in our planet, but they need time. They need time. Something wrong, they die, and something right, they go. And somebody maybe talk about how scientists can identify the age of the egg. So I also discuss about this. So the most used technology to identify the age of something is radiometric dating. So for example, we talk about the rock, the rock. So with like the fossil or the rock. So scientists ask, how old this one? So from beginning. So we have some isotope in here, like the yellow, like the 100% that in the concentration of this one in the density, yes, the density of this isotope, the yellow one, is in the 100% at the beginning, and then through the time, the isotope is degrade and it tries to another chemical, another type item, another type item, and they're looking for the the time and synchronize with the concentration of this isotope in the density of this isotope, and they can infer the time of the subject. So the less concentration density of the isotope in the rock, so the longer time of the rocks. Yeah. So based on this one, so all of the stuff is the scientists is estimate the age of the stuff. So but the chemical in here, they're not just only one. Right now, the scientists can identify a lot of isotopes. They have a better indicator for the age. And this is just some picture I found in the internet, like some kind of something maybe changed because of that kind of event we don't know yet. And the scientists try to explain for that. Okay. this is this figure show like a multicellular creature, like multicellular stuff in about 4 billion years ago. This is about the slide is maybe not true, but this is maybe new finding. We still don't know. So they did the fossils, so like any layer of the earth will be covered different types of the true the event in the earth. So if we dig deeper and deeper, the more information we know about our planet and the living thing ever happened to the planet. So you can follow this one to see the explanation for that. Okay, that is we go back to the diversity of the cells. Like cell come in a variety of the shape, size, and function. Like we have nerve cells, we have some bacteria, we have yeast cells, and we have the active bacteria in your gut may harmful for the cancer. So you see, the size of the cell is very different. Small one to the very big one, 100 times different. But the function of also different. But we believe all we accept that all the cells may be from the same ancestor cells a long time ago, and they diverge after the evolution. And that's a single cell. This is all living organism is controlled by cells. You see, we have a single cells, and we have the insect, plant, and animal. There is a multi-cellular creature. So all of the stuff is making this from single cells. Single cell. So cell is therefore is a fundamental unit of the life because all the cells have the same structure. I mean, there's some different, some subtle different, but the structure is quite the same. But how about the virus? Virus is a cell because they also have the membrane, right? But they have also the membrane, and they have the nucleic acids for genetic transfer, like RNA, double RNA, single RNA, or single DNA, or double DNA. But actually, the virus is not a living thing. Virus like jump by or something. Because what? Because the virus cannot live in themselves. They have to attack to the living cell and use some kind of stuff in the cells, like molecular energy in that cell to live in. So virus not independent life. And we talk more close is human. So all of the part of our body is made up by the cells. See? So we have the first cell, very first cell. That first cell is to transform to become muscle cell, blood cell, nerve cells, and other functional cells. So you see, the cell is quite complex in here. And this different of the cell type also have a different function, different organization, but they come from the very first stem cell, the zygote. And all the cells have almost similar basic chemistry. They have the DNA, they have the DNA for storing the genetic information, they have RNA to transmit that genetic information to the functional molecule in the protein by translation, and the diverse of a protein to make the work. So all of the stuff need protein, from bacteria to animal, plant, anything. They need protein. They need DNA. They have also they need RNA. So all of the stem cells make from the same cell long time ago, the ancestral cells. And the gene is the store information and direct another work of uh, of the cells. But the thing is, we have some discussion. DNA not the first, as you see here, DNA to the RNA to the protein. But a lot of scientists accept that DNA is not the first molecule in this tree. We have another molecule. So this is you already know. I just briefly talked. So discovery of the cells. We have this is Robert Hooke, the England scientist. He identified he making microscope. He looked in the plant tissue and see some box in there, and he named that is the cells box. But the man is looking for the living cells. So this one, the last one is the best cell. This one is Anthony van Leeuwenhoek. He made the microscope himself and looking for the drop of the water, and you see some living stuff in the cells. So you can also make this one and see the cell like him. After that, he writes some stuff. He writes a book and describe the word under the eyes to the community can understand about some micro stuff. And right now in the lab, in the institute, in the company, we not use that kind of thing. We use a line microscope more complex and more power. Let's say this line microscope, a structural line in here. And you see these are same cells in the same cell. But the microscope can generate different picture. You see more clear in different angle of the cell, like here. You see the more surface of the cell, and this is the more intersects of the cells. This is cell nucleus and another. So I have a 10-year working with the cells and looking the cell every day and see different cells have different shape, and they have similar. They have a similar color like this, like gray stuff. Because the light is enter and pass through the cells, you can easily look at the cell like this. So how the scientists can looking for the organelles of the cell? First is nucleus. So we have to send the cell by the dye, and the dye specific dye to bind to some specific molecules inside of the cells, and we can recognize it, the organelles of the cell. Here, we look in the left side is we have first teeth gland, and this is the kidney animal. So all we have the nucleus, like the pin here, because we have specific dye. And now we have a very specific dye. It's better dye. Is that we have a fluorescence. So the resolution of the picture is more, so it's a little bit better. So we have a fluorescent microscope. The microscope can see different type of color, and we have a better cell, a better picture of the cell, more detail of the cell. And here you can see that not only we see the nucleus, we can see the spindle of the chromosome when the cells start to divide. And we have the transmission electron microscope 10. So in here we can see very detailed the cell. Here is the best technology to see the picture of the cells. More inside the cell. We have this is the ER, and this is the dot like the ribosome. Very small. We can see some small organelles, some part of the cells. And we have another one is scanning, different with the last method 10, transmission electron microscope. This is we have scanning electron microscope. So the picture of the scanning microscope is very specific. They can see the surface of the cells. Okay. This is quite a hair cells. So we can see the outside of the cells with SAM, and with SAM we can see inside of the cells. And I also very like this image. So they will show you very detailed from the scale from big to small. We have the finger, your fingerprint, and the cells under your skin. And in here is the heat cells, and this is the mitochondria. In the mitochondria, we have the ribosome working, making protein in there. And here we have the single we have single ribosome, and we can we see the protein of ribosome, any kind of molecules or even item? We cannot use microscope to look this level. From the ribosome we can see it, but from the molecule we can't see it. How can we see that microscope? Can we see that? The answer is yes. So we use a crystallography. If we want to look the structure of the protein. So this technology is the mechanism is also simple. Like you see, if you put a protein in the liquid, the protein with the heat out in the environment, the protein is moving around, right? It's moving around in the chaotic there. So you have to make the crystal, make the protein in the hard form, like the rock, and all the protein, all the molecules of the protein will be stopped. They're not moving anymore. So we use the X-ray. And we track the protein structure, and you see any figure of the protein. And based on this one, and the computer will help you to establish the structure of the protein. This is the most power method to looking for the structure of the protein. And from here, and from electron density map, you can see any kind of the atom in the cells. No, sorry, in the protein, in the molecule structure. Okay, so this is some linked a file and help you to get some different form of cells, from simple to the complex thing. But all of them is a single cell, single cell.